Hello to all of my friends joining us via our recording. Today is March 24th. We are going to start talking about lesson number 9, start talking about material related to the muscles. So to get us started, I am going to ask a few general questions. I'll probably start most of my office hours with these questions. Pop up the first one here. Let me get you a poll. The way that polls work, for my friends who are this is your first time in office hours, you should see a little graph that popped up underneath this question. Click on your answer for me. That helps me know what day you used to come to class with me. So when did you used to have normal lecture time with me? And by the way, if you don't see a little pop-up window that lets you pick an answer, click on the picture of the graph that's next to our friend waving their hand. That should open up a thing where you can pick your answer to the question. About half of us have told me when you come to class. Okay. Yeah, can you see, Eileen, can you see the thing to vote in it too? Let's see if we... Perfect. Okay, so I got a lot of us voting in there. By the way, I'll mention that these are completely anonymous. I have no idea how to see the answers. So with some of the questions that are coming up here, I have no idea who voted what. So totally be honest with me. That's gonna help our time here be most productive. So I'll show you guys what we got going on. We are pretty well split. Let me go ahead. I'm gonna mute everybody. Do me a favor too. I mentioned this before. Notice underneath the poll that you're seeing and underneath the question, just make sure to always leave your microphone with a line through it, unless you are asking a question specifically. So that'll help us with our sound quality today. So I've got friends here from every class, which is perfect. Help me out. If there's anything that we say during our class today that you feel is really important, please do push that out using GroupMe. As always, I'm going to record this session. I'm going to post it for everyone to get to. But if there's anything that you feel like would be really good for your classmates to know right away, please use GroupMe for that. Send that out to let everyone know. So we are polled in there. So I'm going to go ahead and close that poll. Friends from every day, that's perfect. My next question is, I want to know if this is your first time. Let me stop the old poll. Your first time attending office hours, or if you've been before. So let me get you a new poll started there. That should pop up for you. First time, or if you are joining me again. We've only had one other office hour, so if it's your first time, you haven't missed much. Although it was 2 hours, so we went the whole time yesterday. Okay, I got about 2/3 of us who voted. Again, don't be afraid. If it's your first time here today, try to find that A button to click on that graph. If it's your second time here, click B for me. Let's see where we're at. All right, so I'll show you guys. We're about half and half. So welcome to my friends that this is your first time at virtual office hours. I'm mainly going to be the one talking, which takes the pressure off of you. You don't have to talk if you don't want to. In the chat, honestly, is probably your best way to talk. But if you have a specific question that you want to say to me out loud, that's totally fine. Methuselah, I see your hand up. Do you want to ask me a question, or did you just bump that? Either you can put it in the chat, or if you want to tap your microphone, that would turn it on, and you can say a question. I just want to make sure that you don't have a question for me with your hand up. Maybe you just bumped it, which is totally fine.
That was a mistake, I guess.
Okay, that's totally fine. I just wanted to make sure. So I'm going to go ahead and lower your hand, but hey, now we all know, right? If you raise your hand, I'm going to call on you. So No pressure though. This is a totally a low pressure environment. So, all right. So, again, for those of us that this is your first time to office hours, welcome. I will be here every day, 9:30 to 11:30. What I told my friends yesterday is if you log in at like 11 o'clock tomorrow and I'm not in here, shoot me a quick email. It means that, I don't think this is likely, but it means that nobody was there for a while, and so I just left the room. But I will still be at my desk available to hop right back on anytime between 9:30 and 11:30. So I'm glad you all found me. I'm glad you're all here today. We're going to get some stuff done today. So I'm going to stop that poll. Another question for us here. Let's see if it'll, there we go. Okay, so this is my last one. This is going to help me know where we're at today. Let me put in your choices for you before we kind of dive in. Here's my poll. I want to know how much your dog likes my voice. Wonderful. I'm glad to hear that. I want to know how much of lesson number 9 you've been able to work on. So that's the muscular system. And again, this is completely anonymous. So if your answer choice is C, that you haven't started working, there's no shame. I have no problem with you telling me that you haven't done anything yet. I don't care. So gives me a feel for where we're at. Okay, about 2/3 of us have voted. Let's see if we got any more votes to come in. How much of lesson number 9 have we worked on? Some people are afraid of me. It's kind of like in class when I would post those poll everywheres. I know I would almost never get everyone to respond. That's the way it is here too. That's okay. All right, so I'll just show you guys where we're at. Most of us have at least started, which is awesome. What I will mention for you, let me switch screens really fast to show you. Hopefully you found this. Let me end my poll here. This is the schedule that I'm going to post for you each week in terms of what we're generally going to work on during office hours each day. So yesterday's office hours for Monday, what we really all we talked about was what it looks like to go online. So if you have questions about where to find things, how to use things, the recording from yesterday's session is posted. It's in the link right underneath what you used to get in today. I mentioned this yesterday, and I will say again, if you look over here, there's a list of things that I recommend you do before you come to a session. You don't have to do these things before you come to the session. So for example, today I recommended that you work on the first half of lesson number 9. If you came today and you didn't have a chance to do that, that's totally fine. It may mean that today you'll feel like you're at a little bit of a disadvantage because you may not quite track with everything we're talking about. But please always come to office hours, even if you haven't had a chance to work on what I recommend. So for today, we're primarily going to focus on the first part of lesson number 9. So the first part of the muscular system. Tomorrow, we're going to focus more on the second part of the muscular system. So for today's lesson, we're primarily going to look at pictures, talk about stuff from the first half of that lesson. Tomorrow, to get you ready, please try to work on that second half of the lesson. That's going to help you get an idea of what's going on, what we're talking about, and give you an idea of the kinds of questions that you have. Thursday, we're going to switch gears a little bit, and we're going to move over to talking about lab stuff. For lab, there is a guided practice activity just like there is for lecture. So please try by Thursday to work your way through that activity to help you complete the lab packet. I will mention that if you go into the Google Drive now, the lab packet is labeled week 11. When you open it up, it'll still say week 10 on the title on the paper because that's what it used to be. But week 11, so nervous tissue and then stuff going on in the legs, that is what's happening this week. So by Thursday, please try to have completed your lab packet and bring your questions with. I'm hoping to maybe pull a skeleton to have in my office so that maybe we can look at some stuff together on the skeleton. So Thursday is primarily going to be the lab day this week. And then I'm also available on Friday just for general stuff. So any questions you still have about lecture or lab from this week, that's going to be the Friday session. What I'll mention for each of these sessions too is you'll notice that the last thing I have on your checklist is to come up with a list of questions. Every day I always ask you for a list of questions because it doesn't make sense for me to lecture at you, especially in a weird environment. So we're going to kind of work through questions. I'm still going to ask you guys, we're going to use the chat for this a lot. I'm still going to ask you guys to give me feedback to help me get the kinds of information we need on our picture. So today, hopefully we've got a few questions. If we don't have a lot of questions for today, that's fine. We will learn how to do that. But this is available for you in the lecture resources folder. Let's see if I can get all technical on you. Sally, I see your hand. I will get you in just a minute. Let me switch screens really fast. Let's go to Chrome. Let's see where we're at. Okay. Here's what you see when you log into our course Blackboard site. Over on the left, the place that we all use to get into office hours today, I'll just mention here's that link that you clicked in. So you already found this one. Right below it, it says virtual office hours recordings. That is how you will find by tonight the recording from this session, and that's where you'll always find recordings. The schedule for office hours is in the lecture resources folder. So I'm going to open up lecture resources. First document, in case you have not had a chance to look at it, is the frequently asked questions. Please go through and read that because that will answer probably a lot of the questions that you might have for me. Full disclosure, you know, medical facilities are triage patients when they come in, right? Most important, least important, we got to deal with you right now or you can wait kind of thing. That's kind of my email inbox right now. So if you send me an email, I kind of have to triage it. If it's something that I answered in frequently asked questions, I may not reply very quickly because I am also getting a lot of emails from AMP faculty, from my department chair that I really have to deal with. So please don't be offended if it takes me a little while to reply to you. Maybe check out frequently asked questions before you send an email just to make sure the answer's not there. Check out GroupMe to make sure the answer's not there, and then you can send me that email. Kind of as a last resort, because I don't want you to be waiting for really important information for too long. But here's that document I mentioned for you, the schedule for virtual office hours this week, telling you what we're going to be talking about, what you should do to prepare. That's right there in lecture resources. So the document that I showed you guys, that's where you'd find it, in lecture resources. All right, let me switch back. What questions, if any, do we have right now? Do we have any general course questions before we start talking about content? Half of us are new to this whole thing too. Maybe we don't have questions. Did I misunderstand what you said about how many times we do the homework? Okay, that's a good question. In the lab is still 3. That is correct. At least as of right now, for the lab homework assignments, just like we did during the regular semester, you get 3 attempts on the lab homework assignments. What has changed from what the frequently asked questions document says is that for lecture assignments, you have unlimited attempts. So at least as of right now, there's a difference between lecture and lab. In lecture, I'm giving you unlimited attempts on your homework assignments. In lab, you will have 3 attempts on your homework assignment. That's a good question. Yes. I'll show you guys too. When I talk about lesson number 9 today, so we're gonna go here in on learning modules. here's where you'll find the lesson to help you work your way through lecture number 9, essentially. So when I say to do pages 1 through 11, I mean on this link, we'll pull it up here in a minute, page numbers. So those page numbers are always from the lessons. Homework assignment down below it is, since I'm not seeing you guys in class, we aren't doing lecture review quizzes anymore. So the only way you can assess your understanding, assess your knowledge, is to use this homework assignment. So that change happened yesterday. You have unlimited attempts on that assignment. Please use those to help you not only get the highest score, but make sure you really understand what's going on. And by the way, when you look at the schedule of things I recommend that you do before Friday's session, one of the things I say is to do the homework assignment at least once, because that'll give you an idea of, okay, which parts of this lesson do I not understand? Which parts am I not getting 100% on? Like I said, you have unlimited attempts now, so do it like 5 times. It really doesn't matter. You can do it as many times as you'd like to get that practice. Let's see. Okay, yeah, so we're transitioning a little bit into the kinds of questions that we have that we want to address today. So one of the things Lean wants to talk about today is the types of channels. Want to make my screen bigger? Yes, I can try to zoom it in a little bit. Let's see. And on your computer, it might be a little bit bigger. I have mine zoomed out a little bit. So online learning modules folder and lecture resources. Here's that lesson that hopefully you've started working on to get ready for today. Here's the homework assignment for the week underneath it. Okay, so let me get a piece of paper for my reference. Let's see if I can find one. I need your guys' help. I said that today was going to be driven a lot by the kinds of questions that you guys have. So I need you guys to, for me in the chat, please give me an idea of some of the things you want me to talk about today. Eileen says she wants to talk about channels. So I'm going to write down, we want to talk about channels. What other things do we want to make sure we talk about today? Because I'm going to make a list here on my desk. And as we talk about them, I'm going to cross them off my list. So help me out. What are some of the topics from the first part of the lesson, or even if you started working on the second part, some things you might want to work on today? What are some of the things we want to cover during our time today? Okay, so yep, excitation, contraction, coupling. That is a process, so we'll talk our way through that process. Definitely, we can do that. Contraction, coupling. And some of us are still trying to find the words to figure out what we want to ask for today. Okay, ATP and ADP, we can definitely, yep, we can talk about the difference. Remind me, Letty, what lecture class did you normally have? Because I'm betting you're in my one class that I didn't get a chance to talk about this with. I'm just curious when your class was. Monday, Wednesday. Yep, that's what I figured. So yes, we can definitely talk about the difference between ATP and ADP. Absolutely. Okay, yep. So yeah, if we have nothing else to talk about, we can absolutely talk about acetylcholine and acetylcholine esterase. That is something too that technically is in the second half of the lesson. So I'm sure I'm going to have time today. We should be able to talk about at least the basics of that. I may not go as far as talking about Botox today. I may make you think a little bit on it, but I think we can definitely talk about acetylcholine and acetylcholine esterase at least a little bit today for sure. Let me pose the question one other way. Are there specific pictures that you guys would like us to talk about in person? Ask my question one other way. If you flip through your lecture outline, or if you have the lesson pulled up on your computer, are there particular pictures that you'd like a little more clarification on? My last question, and then we'll start talking about some stuff. By the way, for my friends who came yesterday, I told you my office was 75. It's at 73 today, so hopefully we stay at 73 and don't get higher. The pregnant lady is like, man, we don't need to be hot doing this. No takers on the picture question. Okay, well, let's see. I'm going to go over to PowerPoint and see if I can find a good picture. Oh, let me zoom out a little bit. Okay, so one of the topics that we said we wanted to talk about today was excitation, contraction, coupling. That is a process. We go through a series of steps. I'm actually going to switch. Let's do a whiteboard here. We can type some stuff and talk about some things. Okay, so what we are going to talk about, big picture, what we're talking about, is called excitation, contraction, coupling. Okay, 7 or 8, and that's 7 or 8 on the lesson. Is that correct? Pictures in lesson. Okay, so we need to first kind of define lab packet. Okay, yeah, if you could do that for me, find where it most lines up with the lesson, that'd be helpful. When we talk about excitation, contraction, coupling, we should probably break down that terminology, because I think some of what's a little bit scary about it is just the words that are in its name. So let's talk about some of these words that we see in the name of excitation, contraction, coupling. Big picture, what excitation, contraction, coupling is. Let's start with contraction. When I talk about contraction, we are talking about muscle contraction. So the goal of this process is to get your muscles to contract. For your muscle to contract, it needs a signal to telling it to contract. So the excitation part of its name is the signal to make, okay, perfect, page 9. Okay. The signal to make a muscle contract. So when I talk about the process of excitation, contraction, coupling, what I'm talking about is taking a signal, taking a signal that my nervous system sends and telling my muscles that it's time for them to contract. So when we think about how to get your muscles to contract, we need two things. First thing we need is a neuron to send a message to that muscle. The way that neurons talk to each other and the way that they talk to muscles when they're trying to tell muscles to contract is by using neurotransmitters. So neurotransmitters are the kind of messages that a neuron will send. So the first thing I need for excitation, contraction, coupling to happen is I need a neuron to send a message. So we start with nervous tissue to get the muscle tissue to work. Once my nervous tissue sends a neurotransmitter and it sends a message, then the second thing that's going to happen is my muscle will contract. So you look at the name of this process: excitation, contraction, coupling. The excitation part of it is what we outlined up here, sending a message that's going to make my muscle cell contract. Contraction is the second part of its name, getting that muscle to contract. Let me pause for a moment. We haven't covered a lot, but before I move on, what questions do we have? Do we have any questions at this point? Pauline's good. Nobody else is typing. Well, somebody might be. Yeah, not so far, right? LOL. I get that. Yep. Okay. Well, then let's expand this a little bit and talk a little bit more then. So, step one, we said that I need my neuron to send a message. Here's where I'm gonna try to get you guys to help me out here. When I say that a neuron is sending a message, there is one kind of message that it sends. There's a particular neurotransmitter that neurons use to talk to muscle cells. Does anyone know the name, or maybe let's do the abbreviation, right? Because it's way too long of a name. Can anyone tell me what kind of neurotransmitter neurons use to talk to muscles? Do we remember its name? Actually, it's something that we wanted to cover today. Somebody mentioned they wanted to cover this topic. An action potential, Pauline, I'll mention for you, is what happens in the muscle cells membrane when it gets this message. Yeah, Isabella identified it for me. The message that a neurotransmitter sends is acetylcholine. Acetylcholine. Let's put that here. ACH. Acetylcholine. That is the name of the chemical message that a neuron sends to a muscle cell to tell it that it is time for the muscle to contract. So neurons spit out this chemical called acetylcholine, and that chemical is going to interact with a muscle cell. So here's where I might pull up some of those pictures we got. Let me go find my picture here. We are going to pull up that picture I started with. Bear with me. Right here. Okay, we're going to look at this picture right now. I don't have the notes outline in front of me. For those of you that do, if you want to type into the chat for your neighbors to help them know what page we're looking at here. Like I said, I don't have it in front of me, but I'm gonna mark up this page 8. Perfect. Okay, so you'll find this picture on page 8. This picture is showing me excitation, contraction, coupling. It's showing me big picture. Oh, 6. Apologies, Eileen meant page 6. So on page 6, you will find this picture. This picture shows me excitation, contraction, coupling. Remember, excitation meant that a neuron is sending a message. So if I look on my picture here, here's a neuron. Hey, I can do this kind of poll really fast. Raise your hand if you have had a chance to start working on the lab packet for this week. How many of us have started to work on nervous tissue? Okay, a couple of people have. Okay, so a lot of us are still working on it. That's totally awesome. We haven't started yet. Let me give you just a brief crash course to help you out. So I'm going to lower my hands to my friends who have started working on it. Thank you for that reference. Okay, When we talk about a neuron, Dr. Allis is going to get all artistic on you right here. Okay. And man, if we were still meeting in person, which I wish we were, going to be honest, I'd rather be with you guys. So if we were meeting in person, you would get real good at drawing neurons here. So I know we all wish we were together. So this is as close as it comes, right? We're sharing the love from a distance. So okay, so if we were in class, we would be drawing a whole lot of these beautiful neurons together. Check out my gorgeous neuron right here. So this is the extent of my artistic abilities. Although that's not totally true, because I can draw a really awesome cartoon penguin. If we have time at the end of our class today, I'll draw you guys a penguin. So here is a neuron. Yeah, Isabella wants the penguin. We'll get the penguin here at the end of class today. So what I would recommend that you guys do is get yourself either a space on your paper or find a blank something. We're going to talk really fast about this beautiful neuron that I got going on over here. So this neuron that we're looking at has some parts that you are labeling in lab this week. Some must-know parts on neurons. So let me switch colors since we're going to label now. So the first part of a neuron that we need to know are these little branches that you see up here. I probably should have made them arrows, right? So here's my little branches that I'm seeing around here. These little branches that are coming off of a neuron, these things are called dendrites. Yeah, Pauline got it, dendrites. Those are my dendrites. Ooh, spelled that wrong. You'll have to bear with me too. Still learning the technology, so let's see how that looks. Ooh, that looks terrible. Try again. Dendrites. Okay. Dendrites are the part of my neuron. I will get your question, Eileen. Give me just a minute. Dendrites are the part of my neuron that receive messages. So I've got a bunch of dendrites. When I look at my picture over here on the left, we cannot see dendrites. There are no dendrites here because this picture, this part that I'm seeing right here, the part that I'm seeing right here, my picture actually matches this part right here. So yeah, tree branches, that's a good way to think about dendrites, absolutely. So when I look at my picture of a neuron here, recognize that they kind of just chopped it. And in this picture, they're like, your dendrites up here, they don't matter. There's this big part of my neuron right here. This big part of my neuron is called the cell body. Again, on the picture that we're looking at right now from your lecture notes, they decided, the cell body doesn't matter. Yeah, or the soma, same name. So we've got this cell body part here, this soma part here. Attached to it are all these little dendrites, these little tree branches. When a neuron decides that it's going to tell a muscle to contract, because that's what we talked about, excitation, a neuron's got to send a message to make a muscle contract. When it decides that the muscle needs to contract, it's going to send a message down this long part that my picture finally shows. This long part right here is called the axon of my neuron. So the axon of the neuron is the big long extension. And again, that's the part we can finally see on our picture over here. So we're seeing the axon over here. The axon is where a neuron sends a message away. In this case, it's sending a message away down to a muscle cell, a muscle fiber, to tell that muscle to contract. But I can't actually talk to a muscle at any place except these little places down here at the end. These little guys down here at the end have a couple of different names. You can pick the name that you like best. The name I like best is the Axon Terminal. The end of the axon. That's what I like to call these little guys, the axon terminals. You can call them, you'll see this in the lab packet as well, the synaptic knob. That's maybe a little bit, or my picture has it labeled the synaptic end bulb. Any of those names work to describe the very end of a neuron. So my neurons, the part that we're focusing on for this lesson, for the muscular system, is basically the parts that we see sending a message away my axon that sends a message away. And we're also focusing on the very end of the axon terminal, where it's able to actually communicate with or touch a muscle cell. So the excitation half of excitation, contraction, coupling. The excitation half is what's going on with a neuron. To help us with the what's going on with a neuron, we've got this nice picture. I'm going to try to zoom in on it here in the bottom. Let's recenter right here. So what I hope you're mostly seeing on your screen is the little second part of the muscle. Let me draw a box. We'll see if we're all in the same area. Everyone see the part that I'm outlining in red? That's focus on right now. The very bottom part of my picture here. Perfect. Okay, so this is zoomed in really close at the end of my neuron. And this is going to be the location where my neuron actually talks to my muscle cell. So to use some terminology that's going to come up in the lab packet this week, we call this location, it's called the Neuromuscular Junction. Neuromuscular Junction. And its name tells me exactly what's going on. Neuro means what kind of cell? I promise it's not a trick question. Neuro would be, this is when in class you would just scream it out at me, right? Yep, neuron. There we go, neurons. Okay, first half of my name. Neuro means that I'm part of the nervous system here. So we're talking specifically about a type of cell called a neuron, which we just drew our beautiful picture of. Second half of its name is muscular. And when I have that muscular part, what kind of tissue are we talking about? I bet we all got this one. I know we can answer this one. At least I hope we can. It's the name of our lecture, right? Yeah, skeletal muscle, right? Skeletal muscle tissue. So we got muscle cells and we got a neuron that are meeting together. We're looking at the neuromuscular junction. I told you guys that the way that a neuron communicates with muscle tissue is using things called acetylcholine. There's that name that we mentioned before, acetylcholine. Here, I'll type out its full name for us again, acetylcholine. I will tell you on assignments and on quizzes, I will give you the name, or on exams, excuse me, I will give you the name of that molecule. I will also give you its abbreviation. So you will see it both ways. As long as you know that ACH and acetylcholine are the same thing, you should be good. So acetylcholine is the message, the form of the message that a neuron uses to communicate with a muscle cell. Now, when I talk about acetylcholine, acetylcholine is a type of chemical. Acetylcholine is a special chemical that neurons spit out that allow them to communicate with a muscle cell. One of the questions that we were wanting to talk about today was the difference between a chemically gated channel and a voltage gated channel. So let me pause for a moment. I want all of us to do our best to try to answer this question. Can you tell me in the chat what the difference between a chemically gated channel is and a voltage gated channel? And I'm going to give us some time here, so please everyone try to chat at least something to me. Here, let me clear my whiteboard here. Let's get a whiteboard. Bear with me. Okay, here's our question that you're answering for me. What is the difference between a chemically gated channel and a voltage gated channel? I want you to chat me whatever you know right now that makes these different. Let's make a list of what we do know about what makes these kind of channels different. And I'm going to give us some time, so ignore what other people put in. Just tell me what you know. We'll work together as a class here to compile a list. Keep them coming. We're doing great. All right, so let's see. I got 1, 2, 3, 4, 5, 6, 7, 8. Awesome. I got 8 answers in. Okay, so let's start organizing the information that you guys have passed along to me. And if you're still typing, please keep typing. I will add your information to our comparison that we're going to make here. So let's start up at the top. One of the first answers that we got is that one has to do with chemicals and one has to do with charge. Absolutely. So let me get my typing out. Hey, by the way, let me just show you guys, in case you didn't notice, check out my little friend on the right side of the screen here. I know it looks real funky because, you know, I can't draw with my mouse, but there's my little penguin for you. So drew you a penguin while you were typing me your answers. Okay, so let's get back to the serious stuff, right? The reason you're actually here, not for penguins, although it could be for penguins, but not today. Okay, one has to do with chemicals, one has to do with charge. A lot of us talked about and honed in on this part of their name right here, gated. Okay, so let's start with gated. When I use the word gated in class, I always ask you, does that mean it's always or not always open? If you were in class right now, what would you tell me? A gated channel, yeah, is not always open, right? Not always open. Both of these channels are gated channels, which means they are not always open. Let's do, yeah, you have the same confused expression as the penguin. I get that. Okay, so we talked about gated channels being not always open. When we talk about channel proteins, let's do what we'd always do in class. Channel proteins do passive transport. My favorite question from class then. If we are doing passive transport, we're going from blank concentration to blank concentration. Can anyone remember our words that we'd say? We are going from, yep, there we go, high to low. Absolutely. Okay, channel proteins take things from high concentration, where there's a lot of them, to a place where there's low concentration. These are gated channels, so they're not always open. That's going to mean that I only take things from high concentration to low concentration when I open those gates. Now, somebody mentioned for me in our chat an analogy that I gave you guys when we were talking about channel proteins, gated channel proteins. The analogy that I gave you guys was to think about channel proteins are like a fire hose. They're like a fire hose, meaning they move lots of stuff. They move things really quickly. So we've got these chemically gated channels and we've got these voltage gated channels. They're not always open. When they open, They move things really fast. They're moving a lot of things. The difference between them that most of us picked up on in the chat, the difference between a chemically gated channel and a voltage gated channel comes down to what opens the gate. So a chemically gated channel versus a voltage gated channel. We said that they're not always open. We're going to use different things to open them. So when I talk about a chemically gated channel, that color, by the way, I apologize. That looks better as an underline instead of word. Okay. A chemically gated channel is a channel that's going to open up when a chemical attaches. That's what its name means, right? A chemically gated channel opens up when a chemical attaches. A voltage gated channel, like you guys told me about, voltage gated channels, that word voltage has to do with charge. So when the charge on my membrane changes, charge changes. When I go from being really negative on the inside to being positive on the inside, that's going to open up a voltage gated channel. Now, both of these are gated channels, so they're not always open. But for both of these, when I open them, I'm going to let in lots of stuff. It's just a matter of when I'm letting things in or kind of how I'm letting things in. So when I talk about the process of excitation, contraction, coupling, when I talk about a neuron talking to a muscle cell to get it to contract, I use channels both that are chemically gated, where I need a chemical key to open my gate, and I use channels that are voltage gated, meaning I use a change in charge to open them up. Now, before we started drawing penguins, before we started answering this question, we were talking about acetylcholine. See if I can spell it. Acetylcholine. We were talking about ACH. I told you that acetylcholine was a neurotransmitter. When I use this word neurotransmitter, which kind of gate would a neurotransmitter open? What do we think? What might a neurotransmitter be an example of? Any ideas? Yep, chemically. A neurotransmitter is a type of chemical. Neurotransmitter is a chemical. Neurotransmitter is a chemical made by neurons. The goal of acetylcholine is for a neuron to open up a chemically gated channel on a muscle cell. So let me go back to the picture we were looking at before. Say goodbye to our friend the penguin. I'll have to try to draw one again later. Pull up my PowerPoint here. Let's go back to that picture from before. Right here. Zoom in. We are talking about, here it is on a picture again, acetylcholine. We are talking about a little chemical. Here's those chemicals right here. See the red dots? Those are my chemicals. We are talking about a chemical that goes down and attaches to these little purple proteins that you see right here. These little purple proteins that we see right here on my, here's my muscle cell. Here's hopefully a fast question. What's the special name that we use to describe a muscle cell? I call a muscle cell a muscle, there we go. Yep, there we go, it's coming in. A muscle cell is called a muscle fiber. Good, okay. Muscle fiber. Okay, so what I'm looking at in the bottom part of my picture right here, this is my muscle fiber, my muscle cell down here. On the outside of my muscle cell, I have what we call chemically gated sodium channels. Here, let me go underneath because I'm going to lose my word. chemically gated sodium channels. These are the places where those neurotransmitter chemicals attach to. So neurotransmitter chemicals are released from a neuron when the neuron wants to tell the muscle fiber to contract. Those chemicals come down and attach to chemically gated sodium channels on the membrane. Here, let's do another lab word here for us. On the sarcolemma membrane of my muscle fiber. So I have a neuron that spits out chemicals. Chemicals come down, they attach to these chemically gated sodium channels that live on the outside of my muscle cell. I have a gated channel and I have just given it the key that opens up that channel. That is what's going to allow me to change my membrane charge. Let me flip one picture and then I'm going to pause for questions here. Again, I don't know for sure what page this picture is on. Hopefully you guys can sort it out in your chat here. This picture shows me those chemically gated, well, actually, I'll circle it. I don't have to write it. Page 7, perfect. Okay, we're on page 7 now. This picture shows me up close and personal all those little chemically gated sodium channels that we were talking about on the previous slide. This shows me what they look like. So I've got my chemical that came in this case it's acetylcholine. It's that chemical messenger. That chemical came in and it attached to my protein channel. When it attaches, when I put the key into my lock on that gate opens up. Now, that gate is a chemically gated channel. In comes my sodium. In comes my sodium. Hey, here's where we get to do Dr. Ollis's favorite thing from the whole semester. Let's see if she can pull this off at all on the mouse. What am I drawing up here? It does, Isabella. Yep, we'll talk about that. Can anyone tell what Dr. Ollis is attempting to draw? I picked the wrong color. My salty banana. Yes, look at that. All right, here's my salty banana cell. And hey, since I'm not like using a whiteboard, I can use a yellow marker. So we'll make it all yellow. Look at that. Okay, here's my salty banana cell. So let's remind ourselves. This cell is represented by a banana. What do I have a lot of inside my banana cell? What's there a lot of inside here? Okay, I got several votes here for potassium. K plus. Ooh, not yellow. Let me change the color. Inside my cell, we have a whole lot of K plus. That's potassium. Banana's really high in potassium. When we talk about the salty banana cell, what's inside a banana is that potassium. But remember, this is that gross banana that is salty. So there are two things on the outside. Yeah, Letty knew where I was going even before I said it, right? Two things on the outside. One of those things is sodium. So sodium's abbreviation is Na plus. Sodium. What is the other half of salt? Yep, Kathleen's chiming in here with our other half of salt. Our other half of salt is chloride. Chloride. Calcium is important in neurons, but not as important here in a typical cell. So we're not really going to talk about calcium. Yeah, we do also have glucose and proteins. Yep, let's. Oh no. Don't go into the corner, Letty. Stay with us. You're good. Let's put our glucose inside the banana. Let's put our proteins too while we're at it. Just to remind ourselves of the kinds of things we got all over the place. Yeah, nobody has to go to the corner, guys. This is just like in class. Feel free to say the wrong answer. There is no shame. So nobody has to go to the corner. And if you go to the corner, there's definitely a penguin there to comfort you. So just imagine a penguin. So here is my salty banana cell. that we just built over here. Okay, so what I just did on this salty banana cell that we can see from my picture over here on the left is I opened up a channel. I have my chemical key that opened the gate. I opened up the channel for sodium. Remember, these proteins are taking things from high concentration to low concentration. So is sodium going to come in, or is sodium going to go out when I open up this channel for it? Which way does sodium want to move when I open a channel for it? Into the cell or out of the cell? What do we think? Okay. 3 of us, 4 of us are guessing. Okay. We are guessing correctly. Sodium wants to go inside. So let me get my little arrow back here. See how sodium was something we said I've got a whole lot of outside. Things are going to go from where there's a lot to where there's a little. If I make a way for sodium to cross the membrane, it's going to rush inside. So when I open up the chemically gated sodium channel, we'll be specific here. It's a sodium channel. Oh, Dr. Allis, why are we writing sodium? Okay, this is a chemically gated sodium channel, which means that sodium wants to come inside. Let's do some terminology review here. Sodium has a positive charge. When I talk about something with a charge, it is called an ion. But when I talk about something with a positive charge, what's my special name for positive charges? There's a special name for those. Yep, there we go. Popping up in our chat for us. A positive charged ion is called a cation. Cations have a positive charge. When I open up my chemically gated sodium channel, because my neurons spit out a chemical called acetylcholine, in rushes my sodium. In comes the sodium that we see right here. Sodium, because it's a cation, has a positive charge. And if I bring in a whole bunch of positives, what will happen to my membrane charge is my membrane charge, or, because here, let me underline for us. It's been a while since we talked about this word. Remember I told you guys way back in Unit 1, if you see this word potential, write the word charge next to it. So if I've got a charge that's normally negative, by the way, this would be an underline highlight star number down here, negative 90 millivolts. Muscle cells are more negative even than neurons. So muscle cells normally are chilling out at negative 90. If I open up a chemically gated sodium channel, they're going to rush in a bunch of sodium. And because sodium has a positive charge, we're going to add positive, positive, positive until our charge goes all the way up to positive 30. That would be another underline highlight star number, positive 30. When I bring in a ton of sodium, that is going to get my membrane charge all the way up to positive 30. Positive 30, by the way, is going to be a charge that's really helpful to get my muscle cell to release calcium. So I'm going to add a little note right here next to positive 30. This leads to calcium, Ca2+, release when we're all the way up at positive 30. Okay, so this would be the point, if we weren't locked in our virtual room, right, that I would now tell you to turn to your neighbors and talk. And it's really hard for us to do that in a classroom like this, right? So instead of that, here's what I want to do. I want to stop talking here for about a minute. I want you to kind of try to think about this process, try to explain it, even maybe out loud, to your dog, to your pillow, to your wall. I want you to try to explain what we just talked about to help you figure out what questions you need to ask me right now. So I'm going to stop talking for some time here. I want you to brainstorm for me a list of questions that we have before we move on. So let me pause and give you some time to process. If you have thought of any questions, or if there's a particular part of this slide you want me to talk about a little bit more, can you post something in the chat for me to help me know what to clarify, if there's anything in particular we're thinking right now? Okay, good. Eileen says that our discussion is helping. Awesome. It's going to take processing. So even if it's just I sort of understand it a little bit better, that's great. It's going to take some processing. Let me ask the question too in another way. If you feel like you're ready for us to move on, to keep talking about stuff, can you raise your hand for me? Can you raise your hand if you think you're ready for us to move on? Okay, Isabella has a question for us. Some of us are still processing. Let's mention Isabella's question. Isabella asked about the membrane potential or the membrane charge. She asked, is it always either -90 or +30? When a muscle is not contracting, when it's not getting a message from a neuron, yes, it is always -90. That is a term. Let me give you a term. If you remember way back from Unit 1, we talked about polarized cells. A polarized cell is a cell that is different inside and outside. So yes, when a muscle is not getting a message to contract, it is always -90. When a muscle gets the message to contract, it will always go all the way up to +30. That's what it does when it gets the message to contract. So the two numbers we want to know on muscle cells are -90 when they're waiting for a message, and +30 when they have received a message and they're going to contract. Like I mentioned, if we were in class, I'd say, I don't normally ask you numbers, but here's the two numbers we need to know: -90 and +30. We need to know those guys for muscle cells. Yes, Wink, wink, nudge, nudge. Yeah, so Eileen asked about when we get up to positive 30, is that what is going to lead to the calcium release? Kind of. I'm going to clarify that a little bit more, but at positive 30, that is when a muscle cell will be spitting out its calcium, and that's going to help that muscle cell to do the process of contraction. Yes. I'm going to, actually, I can do it right here in the PowerPoint. Let me go find it. I was looking at the slide I want to talk with you about, actually one more, right here. Help me out. I know you guys have this picture in your somewhere. Let's help our neighbors. What page of your notes packet do you have this 3-panel on? Page 8. Perfect. Okay, so let's transition over to page 8. Here's what we've talked about so far. Up here in the top, we are looking at a neuron. This is the part down here at the bottom. Remember that we were calling the axon terminal. The axon terminal. That's the end of the road for the neuron. So at the axon terminal, that's where I'm going to meet my muscle cell to make contraction happen. At the axon terminal, see all these little green dots down here? All these little green dots that were right here, these are acetylcholine. We're going to abbreviate. Good guess, Isabella. They do kind of look like ribosomes, don't they? But they're living on the outside of a cell. No, you're totally fine. Yeah, there's no harm in saying an answer that's not correct. No judgment zone, right? We got lots of penguins here. There's no judgment here. So here's my little acetylcholine. What we've talked about so far, and here let's put our word on it that we said before. What we're looking at in my top frame up here is excitation. Excitation, when my neuron sends a message to a muscle cell. So I've got the axon terminal, which is the end of that neuron. At the end of that neuron, it spits out its acetylcholine messages. Those messages go down, like we just looked at on our previous slide. They go down and attach to these little channels right here. So see my little purple channel that's got the green dot on it? We'll add a label to it. Those channels that we just talked about are chemically gated sodium channels. Chemically gated sodium channels. Let me add my arrow down to them. Okay. My neuron spit out a chemical called acetylcholine. That chemical attached to my chemically gated sodium channels. My membrane charge went up to positive 30. So let's make a note. When I open up my chemically gated sodium channels, membrane charge gets to positive 30 millivolts. Because I opened up those channels, my membrane charge gets to positive 30. That's what we've talked about so far. Now we need to tie together what's happening at this one place, because my picture over here, this shows me one place on the entire membrane of my muscle cell. I can't just have one place on my muscle cell's membrane depolarize. I can't just have one place where I go to positive 30. Everywhere has to go to positive 30 for me to be able to get my muscle cell to contract. So here's a terminology review for us. Here's a word we need to know. Action potential. Action potential. An action potential is a change in membrane charge. Let me go underneath it. Moves. A change in membrane charge that moves. It is no good for me if the only place on my membrane of a muscle cell that depolarizes is just right by the neuron. I need my entire muscle cell to depolarize so that that whole cell can contract. So I'm going to use what's called an action potential to get the entire membrane to change charge. And action potentials are the reason why, if you remember way back to before spring break, before the world went crazy, right? Way back before spring break, we talked about some structures called T-tubules. Let's start these guys here. T-tubules. Let's see if we can answer this question. I think you guys can answer this question. What are T-tubules? What do we remember from lab about what T-tubules are? What are those things? Any ideas? Not quite blood vessels. The blue part in my picture is the sarcoplasmiculum. I do see T-tubules in this picture. I might actually bounce to a different screen to see them better. Here's a T-tubule right here. When I talk about T-tubules, these are places where the plasma membrane of a muscle cell folds in on itself. Okay, so I'm about to switch slides, which sadly means I'm going to lose all of these beautiful markings here. If you want to take a quick snapshot of the stuff that we've written here, when I come back, it's going to be gone. So let me give you a minute here if you want to snap it with your phone. It's going to go away on me here because I'm going to go to a different slide to see those T-tubules better. That's okay, Pauline. I'll show you on an image where they look a little bit better. So it's hard to see them on here. Okay, I'm going to flip to another slide. The slide I'm going to go to is right here. Yep, right here. We already saw this picture over here, so we'll tie it in. But right now we're trying to figure out what T-tubules are. So if you check out here on this upper picture, when I talk about T-tubules, the best way to describe what a T-tubule is it's a place where the sarcolemma folds inside. Inside, we'll say the cell, just for completeness, inside the cell. A T-tubule is the place where the sarcolemma folds inside the cell. Remember that word sarcolemma, we see it labeled on our picture up here. The sarcolemma was the fancy anatomy word for the plasma membrane on a skeletal muscle cell. So the sarcolemma is this gray stuff here on the very outside of my muscle cell. When I talk about T-tubules, I'm talking about these little gray tubes that I see right here, T-tubules. These little gray tubes inside a muscle fiber, they are places where the plasma membrane folds into a tube. That tube is continuous with, or it's completely attached to, the sarcolemma. When I change the charge on the sarcolemma, so let's pretend, here I'll draw for us, let's pretend on my picture down here that I have an axon terminal right here. Let's pretend that I have an axon terminal down here. So my axon terminal, like we just talked about, this is where the excitation side of things happens. So we spit out of my axon terminal here, I was spitting out some acetylcholine. I spit out some acetylcholine. That acetylcholine goes down and it depolarizes. Ooh, my mouse moved here. It opens up the chemically gated channels on this part of my membrane right here. That is all well and good, but the problem is when my muscle cell contracts, I don't just need this part right next to the neuromuscular junction to contract. I need my myofibrils that live all the way over here to contract. And even though it's not shown in my picture here, my myofibril tubes are going to go all the way down. They're going to keep going to the very edge of my whiteboard and beyond. I need all parts of my muscle fiber to contract at the same time. So everybody's got to get this message that we depolarized. So what I do is I take that area, my neuromuscular junction, where my neuron interacted with the muscle cell, and I put, let's do some typing here. Remember, I put chemically gated sodium channels right there by the neuron, right here at my neuromuscular junction. But then I cover the rest of the sarcolemma, and I cover these T-tubules covered with voltage gated sodium channels. Voltage gated sodium channels, like we outlined, it means that a change in charge is going to make these guys open up. So the way I get my charge to be different at the beginning is because of my chemicals. I use these chemically gated sodium channels at the beginning when I spit out acetylcholine. They change the charge on one small place of the membrane. Once that small place changes its charge, all of the neighboring places on the membrane are covered in voltage gated channels. So if I freak out one part of the membrane, suddenly the parts that live right next door, they get freaked out too. And they get freaked out, which then freaks out their neighbors. We start with a little positive 30 in the middle, which becomes positive 30 next door, which becomes positive 30 next door, which at some point you're going to bump into these T-tubule things. When you bump into the T-tubules, now I'm changing my membrane charge going down into the cell, not just on the outside on the sarcolemma. Now I'm changing my charge inside the cell. So that brings us to the part of the picture. Let me get my marker here. That brings us to right here. This space here in the middle is my T-tubule. My T-tubules go down inside a muscle cell. When these T-tubules start to change their plasma membrane charge, they open up and sodium rushes inside. When sodium rushes inside and depolarizes the membrane inside a T-tubule, here's our last important kind of proteins that we see right here. See these guys right here? I know they're small. I'm going to try to circle them here. These small proteins that we see right here, notice that they're called calcium release channels. Or the term that we're going to use to describe these channels, these are mechanically gated calcium. Oh, that's not going to fit. Calcium channels. Draw an arrow to make sure we can find them. We have not talked about mechanically gated channels yet today. We've talked about them in the past. When I talk about something that's mechanically gated, what I want you to think about is the way I open that is I push it open. A mechanically gated channel gets pushed open. So when we're talking about the channels that I use to spit calcium out of my sarcoplasmic reticulum, there's a lab word for us, the kind of channels that open up to let us spit out calcium, they're mechanically gated. You push on them and they open up. The way that I get these to be pushed open is because I've got voltage sensitive or voltage gated proteins, let me get my pointer back, that live right next door to them inside these T-tubules. And these proteins that live right next door to my mechanically gated calcium channels, when they change their shape because the membrane charge is wrong, that pushes on my mechanically gated calcium channels, and then those channels open up. And that leads to the spitting out of calcium. These are big ideas. Again, I really wish we were together so that you could turn to a neighbor and chat this out. I want you to try to either chat it out in the chat box with me, or try to write it out on your paper. Here, let me give you a question to use as your prompt here. Let's see. My question is, what is the role of voltage-gated sodium, go down the line, channels, and well, let's start with that, in depolarizing the whole membrane? There's our first question. And then I'm going to put my second question down in the bottom corner here. How do mechanically gated calcium channels open up? Here's my two questions for you. I want you to try to put some writing in your notes here. My first question, let me circle it up here at the top. What is the role of voltage-gated sodium channels in depolarizing the whole membrane? So these are the kind of channels especially that I find on the T-tubules. I'll put a note here, T-tubules. And your second question, how do I open up those mechanically gated calcium channels? Let's try to write some notes, and I'll stop talking for a bit. Let's see what we can come up with these. And as you are processing, if you think of questions or something that you can put to words that you want to ask me or something you want to clarify, go ahead and pop it into the chat for me. I'll still give us another couple of minutes here because this is the big stuff. I am about to switch slides. So if you want to snap a picture of what you see on the screen, if you want to have a copy of it, all my writing is going to go away. So let me give you a minute here if you want to snap a picture on your phone, because I want to reference another picture here for us. I want to reference this picture right here. Help me out. What page of your notes packet do we see this picture right here? Can anyone tell me what page this picture is from? Page 7. Perfect. Okay, so you have this picture on page 7. Here is what I want to highlight on page 7. Let me get my typing out here. Letty, I see you raised your hand. Do you have a particular question that you wanted to ask, or was it accidental raised hand? Oh, that's totally fine. I just wanted to make sure. Okay. What this page is showing you, circle it right here. Action potential. Action potential. Action potential. A definition that we must know is that an action potential is and we already defined this once, a change in membrane charge that moves. An action potential is a change in membrane charge that moves. What I want you to picture this little line as right here, for lesson number 9, we want to pretend that this line is a T tubule. So what happens is at the beginning of my T tubule, because I depolarized the neuromuscular junction, because those chemicals opened up the channel, that allowed a bunch of sodium to rush inside. We have depolarized part of the T tubule. But remember that we said that all of the T tubule is covered in voltage-gated sodium channels. So if I depolarize the beginning of the T tubule, I bring in a bunch of sodium, this area gets positive. And the voltage-gated sodium channels next door, where my pointer is now, where it's still red, they say, oh my gosh, we're getting positive. I need to open up too. So in my next frame, really short time later, T means time, really short time later, the voltage-gated sodium channels right here are going to open up as well, and in comes the sodium. Well, now this part of my T tubule has a positive charge on it, which means that the neighboring parts next door that have a negative charge right now, they start to realize, oh, we're going positive too. So we go down a little bit longer in time, and look at that. I've opened up my voltage-gated sodium channels here as well. So big idea. Let me get out my typing here. Voltage-gated sodium channels help action potentials move. That's kind of a big picture. If we were in class, I would tell you to write that down and underline, highlight, star it. When you are looking at this picture, when we're talking about an action potential, which means the charge change is moving, the way I'm going to get that change to move is I'm using those voltage-gated channels. So voltage-gated channels take it all the way down the T tubule to allow all the parts of my membrane to depolarize. Everybody freaks out, goes from negative to positive. That's what's going on with an action potential. Again, I know my chat environment is bad for this, but If you have any questions right now, can you pop them in for me? If you have something you want to clarify with me. Any thoughts? You'll be replaying this one. Yeah. I mentioned it at the beginning that I will get the recording of today reposted by the end of day today. As soon as Blackboard and YouTube process it, I will get that posted up for you, so we can definitely replay it. I had a request on the action potential thing. Yep, so I'll describe it for you one more time. Remember, when we talk about an action potential, this is our definition that we want to make sure we know. An action potential is a change in the membrane charge that moves. So when we looked at the charge on a muscle cell, it's always at -90 unless it gets a message from a neuron saying it's time to contract. If it gets a message that says it's time to contract, it's going to go all the way up to +30. I need to get to +30 to be able to contract my muscle cell. So everywhere along my membrane has to get to +30. The way I do that is by changing the charge on everybody. So an action potential is where I freak out the charge on various parts of my membrane. So we said that we're imagining that this is my T-tubule where I fold it in the membrane. The first part, we get positive because of those chemically gated sodium channels at the neuromuscular junction. That brings in a bunch of positive sodium. When I bring in a bunch of positive sodium right here, next door neighbor part says, oh, we're going positive over there. I have voltage gated channels. My channels open up too, and in rushes sodium right here. Now that I've got a bunch of sodium in this section of my T-tubule, this neighboring section next door says, oh, we're starting to go positive. I need to open up my voltage-gated sodium channels as well. So that brings us to the last frame of our picture here. An action potential is when I take that positive charge. Yes, exactly. I do a chain reaction with that positive charge. The first part becomes positive. Once the first part becomes positive, that makes the second part become positive. And once the second part has become positive, then I'm going to make that third part become positive. Every part of my membrane is going to go positive because of these voltage-gated channels. So I think somebody asked, is it technically the action potential that's moving down the membrane, the T-tubules? Yes. And all an action potential is it's just that change in membrane charge that's moving. So yes, the action potential is what goes across the membrane. We go from -90 up to +30 everywhere. It travels down the tube. During lecture really helped in preparing for the test. I lost my message. Just go read it. Correct. Yes. So the new outlines seem, well, I haven't posted new outlines yet. I have not made the new ones. So all you're seeing are the lecture outlines that we had from before. We're still to the point that we do not want to print number 10 and number 11 because I have not posted updated ones. So what I said in yesterday's meeting that we had, our virtual office hours, was that when you're looking at the lessons and you see a page that I've written a bunch of text for you, I want you to pretend like that's what I'm standing in front of the class saying to you out loud. Whatever you would write down if I was saying that to you out loud in person, that's what you want to write down from there. So you are not getting more information than you would have gotten in class, but instead of me saying the information to you, I have it in written form for you. So maybe that helps you appreciate how much I said in class that you didn't write down, because that's all I did, is I took what I would have been saying to you out loud and I put it in words. So you don't have to memorize every single thing, every single word I say. Pretend like I'm saying it and summarize it. Sally asked if my action potential is always when it goes positive. Yes, it is. An action potential is always a change from being negative, because all my cells start at negative all the way up to positive. So yes, an action potential is always going to be where it goes positive. Are the first two on this diagram chemically gated and the last one voltage gated? We can't say for sure exactly which kinds of channels we're looking at on this simplified sketch. What I will say, let me change slides here. Let's figure out what slide to go to that would be most helpful. What I will say is, excuse me, when we look at this picture right here, which we discussed a little while ago, when we look at this picture right here, we can definitely say, beyond a shadow of a doubt, that these little guys right here are chemically gated channels. My channels that live at the neuromuscular junction, they're absolutely chemically gated. When I'm talking about the channels that live next door, the ones that live over here in the sarcolemma, or when I talk about the ones that live in the T tubules, like I see over here, these ones are absolutely going to be voltage gated, because I don't have any neurotransmitters being released there. So I can't say for sure on that picture if the first part of where sodium came in is because of chemically gated or voltage gated channels on that picture in particular. But when you're thinking about it in big picture, when we're thinking about a neuron talking to a muscle, and then that charge going from where it talked on the muscle to other places, everywhere else except where the neuron and the muscle meet, everywhere else is voltage gated. So I will put it this way. I'm not going to ask you to distinguish on that particular picture when you're doing an exam. I'm not going to ask you, okay, this first area where sodium comes in, is that showing me voltage gated or chemically gated? I'm not going to ask that question. So I might ask you if we're at the neuromuscular junction, who lives there, but I'm not going to ask you that specific from that picture. We just want to know, in the words that Eileen used, we want to know that it's a chain reaction. That's a great way to think about it. Does the action potential start at the sarcolemma or the motor end plate? Terminology, motor end plate. Let's clarify this. Motor end plate, you'll see that or hear that in some places. That's the same thing as the neuromuscular junction. Neuromuscular junction. An action potential will start at the neuromuscular junction, or at least my change in membrane's charge is going to start at the neuromuscular junction, because that's the place where a neuron spits out acetylcholine, and that opens up the chemically gated channels. So yes, this whole process always starts at the neuromuscular junction. That is correct. Something I am not going to have a lot of time to discuss today, and nobody really asked us about it at the beginning. We have a really great video for it, so I think that's why. Let me just mention something you do need to know is why I need calcium to be spit out. Let me see if I have a good picture for this. Not really. We'll do this one right here. This one doesn't show me calcium, but what I will mention is the reason that calcium is important. We'll put a note up here. Calcium attaches to one of our regulatory proteins that helps us decide when a muscle contracts. Can anyone remember from your lecture studying or your lab studying? Calcium attaches to which one? Calcium attaches to, yeah, I got a couple of votes come in here. Yep, the one called troponin. Calcium attaches to troponin. The reason that I need my action potential to cause my sarcoplasmic reticulum to spit out calcium is for the sake of troponin. So Troponin, I can see in my picture right here. Troponin. Troponin, I said in lab, I like to think about troponin kind of like a push pin. Troponin is a push pin that helps to hold a protein called tropomyosin in place. Tropomyosin's job is to block the myosin binding sites on actin. When I block those myosin binding sites, no muscle contraction can happen. So when we talk about wanting muscle contraction to happen, the ability for it to happen, first thing that I have to do for muscle contraction, number one, is I got to get troponin out of the way. Troponin is my push pin that's holding tropomyosin in place. If my push pin changes shape because calcium attaches to it, then number two, what's going to happen is tropomyosin is no longer going to be able to stay in place. What I told my lab students is think about tropomyosin like a wet spaghetti noodle. It does not stay covering these myosin binding sites unless it has a push pin, that push pin being troponin, holding it in place. So first thing that happens is calcium comes and attaches to my push pin. My push pin changes shape. If my push pin changes shape, my wet spaghetti noodle, tropomyosin, changes shape as well. If tropomyosin changes shape, it is not blocking those myosin binding sites anymore. And if it's not blocking the myosin binding sites anymore, we get into the process that you see here from your notes. Here's those pictures from your notes called the cross-bridge cycle. So I don't have time to go through the cross-bridge cycle. I do want, because I got a request, I do want to talk about the difference between ATP and ADP. But what I will say with this cross-bridge cycle thing is please make sure you know what's happening in each stage. Really great video that some of us watched in class already. Some of us are watching for the first time at home, but a really great video that walks you through the cross-bridge cycle. So what you need to be thinking of, big picture. Let me go all the way back. Yeah, the rigor mortis video was definitely helpful related to this as well. Big picture, let me switch slides on you, and then we'll talk ADP, ATP. Ultimately, your goal with the first half of lesson number 9 is you need to be able to walk through the process from a neuron sending a message, so a neuron releasing acetylcholine, down to the process of the cross-bridge cycle, how a muscle actually contracts. We need to be able to explain why things have to happen the way that they do. So we just briefly talked about why I need calcium to spit out. All of this action potential stuff that starts here at the neuromuscular junction and continues down through the T-tubules, down to the sarcoplasmic reticulum, all of that, we have to do all of that to get the calcium to spit out. Because if calcium doesn't spit out, I can't do the cross-bridge cycle. So try to think big picture. And yes, there is an exercise in the, I think it is page 11 of the lesson that gets you to try to put those steps in order. That takes you from when a neuron sends a message all the way through the cross-bridge cycle. Yes, we want to be thinking big picture, all these steps. What am I using them for? Why am I doing them? So this is kind of the last image, this slide that I have up right here. This is kind of the big picture, everything we've been talking about today. This is from why the neuron sends a message all the way down to muscle contraction. That's what we were talking about today. Briefly, let me pull up a whiteboard because the last question that I have not covered today is ATP versus ADP. Some of you guys, we got to have this discussion in class, so you can zone out a little bit or you can remind yourself, but I have a chunk of you that we didn't get to have this discussion together. So I want to make sure that we clarify this. You will see in the process of muscle contraction a discussion of ATP and a discussion of ADP. So let's start with ATP. ATP stands for adenosine, that's the A. T is for tri, And P is for phosphate. Adenosine triphosphate. That's what ATP stands for. When I talk about what ATP is, we always said in lecture that that is the form of energy that a muscle cell uses. So let's talk about what ATP looks like. We have our adenosine group, and then we have phosphates that are attached to it. PO4 is the chemistry of what a phosphate actually is. When I talk about adenosine triphosphate, I am talking about this adenosine molecule that's attached to 3 phosphate groups. We got 1, we got 2, we got 3. Adenosine triphosphate is what your body uses to transfer energy. The energy of ATP is right here. This is why I made this bond yellow. This bond here on the outside, the one that connects my second phosphate and my third phosphate, that holds them together, this is where I have energy. So when you have a molecule called ATP, I have an adenosine group that has 1, 2, and 3. Sorry, let me get my pointer out. 1, 2, and 3 phosphate groups. So this little bond right here in yellow, that's where the energy is. When I need to use energy to do something like reactivating, or the word in the video is cocking, the myosin head to get it ready to do the process of muscle contraction again, what I can actually do is chop up this bond. I can cut it, and that will give me some energy to do what I need to do to change my shape. When I chop up this bond, I make what's called adenosine diphosphate. ADP, adenosine diphosphate. So the way that this works is I've got an adenosine group. Attached to that adenosine, I still have 2 phosphates, but I broke the bond between that second one and that third one. So here's my adenosine group still. I have 1 phosphate still attached. I have 2 phosphates still attached. This little phosphate over here, I chopped it off. There's no attachment site between them. So you will see on your picture that they will list this that we're looking at right here. They'll list it as ADP plus PI. PI just means inorganic phosphate. I promise you I will not ask you that on an exam or an assignment. But what happens is I have this molecule up here, ATP, that we started with. It has 3 phosphate groups that are all attached to adenosine. If I break the attachment right here, my yellow attachment, if I chop that up, I still have an adenosine that is attached to 2 phosphate groups. Since it's only attached to 2, now I call it diphosphate. And I have this random phosphate group that has no energy, that's just chilling out. That's my inorganic phosphate. So that's why you'll see the PI thing. It's this random one that's no longer connected. It's not useful for me. So for my friend who asked about ATP and ADP, how they're different, there's a little bit of a crash course there and some chemistry for you about what makes them different. Yes, you're welcome. All right. I am unfortunately, sorry, not going to have time to talk about acetylcholine and acetylcholine esterase today, but I promise we will talk about that tomorrow because that is one of the first topics that pops up on page 11 in your lesson, or page 12, I think, in your lesson. So we will definitely talk about acetylcholine tomorrow, talk about acetylcholine esterase. Let me check something really fast. I made you guys a meme that you'll find in your next set of, let me peek and see if I put that meme in my PowerPoint. Otherwise, you'll just run into it. Yeah, I don't think I saved my meme. So, oh no, I did. Here we go. So here's a teaser for you of what is to come tomorrow. we're going to talk a little bit about Botox. So deadly bacterial toxins in your face, tons of fun. So that will be tomorrow. The rest of lesson number 9, please work your way through that. Please bring your topics you want to talk about. We'll do the same thing tomorrow, same time, same place. So, oh, thank you, Eileen. I appreciate that. I'm glad to be able to help you guys in the ways that I can. So please work your way through the rest of lesson number 9. For any of my friends who are in, let's see, we're Tuesday today. Any friends in Wednesday, Thursday, or Friday labs, please make sure you have completed week number 8 homework. Get it done from lab. Hopefully you've already gotten it done. If you have not, we got to get on that snappy because you also have week number 11 lab homework due on Sunday, everybody. So please make sure that you're knocking out week number 8. Get that muscle stuff done. There's no reason for it to hang over your head. So wrap that up. I know I wish there were more people here for questions too. So talk it up in the group me, right? Tell your friends that it's worth their time. And like I mentioned, I'm going to do my best to get this video recording up to you guys for this evening for any friends who couldn't attend today. Once Blackboard processes it, I'll shoot it through YouTube and it'll be available just like yesterday's was. So if you like this way of teaching, well, good. I'm glad it's working for you. So I'm not quite sure. I'll wait. Martha, your comment came through a little bit funky. So I'll wait to see if you can clarify for me if you want me to return to something. But yeah, I will stick around a little bit longer for questions, but I'm going to go ahead and end our recording now. So I will see you guys, hopefully most of you, again tomorrow morning at 9:30.